And welcome to the video lecture for Unit 5, Module 10.1.4. And this one we're going to discuss the stomach. As always, here are your learning objectives, so be sure to utilize these when studying for both your lecture quiz and the unit exam. Let's go ahead and get started. The stomach has four major functions. 1. Storage of ingested food. 2. Mechanical breakdown of ingested food. 3. Disruption of chemical bonds in food through the action of acids and enzymes. And 4. Production of intrinsic factor, which is a glycoprotein needed for the absorption of vitamin B12 in the small intestines. Ingested substances combine with the secretions of the glands of the stomach, producing a viscous, highly acidic, soupy mixture of partially digested food called chyme. The stomach has the shape of an expanded J. A short, lesser curvature forms the medial surface of the organ, and a long, greater curvature forms the lateral surface. The anterior and posterior surfaces are smoothly rounded. The shape and size of the stomach can vary greatly from individual to individual, and even from one meal to the next. In an average stomach, the lesser curvature is approximately 10 centimeters long, and the greater curvature measures about 40 centimeters. Typically, the stomach extends between the levels of vertebrae T7 and L3. We divide the stomach into four regions. One is the cardia. The cardia is the smallest part of the stomach. It consists of the superior and medial portion of the stomach within 3 centimeters of the junction between the stomach and the esophagus. The cardia contains abundant mucus glands. Their secretions coat the connection with the esophagus and help protect that tube from the acid and enzymes on the stomach. 2. The fundus. The fundus is the portion of the stomach that is superior to the junction between the stomach and the esophagus. The fundus contacts the inferior posterior surface of the diaphragm. 3. The body. The area of the stomach between the fundus and the curve of the J is the body, the largest region of the stomach. The body acts as a mixing tank for ingested food and secretions produced in the stomach. Gastric glands of the fundus and body secrete most of the acids and enzymes involved in gastric digestion. And 4. The pylorus. The pylorus forms the sharp curve of the J. The pylorus is divided into pyloric antrum, which is connected to the body, and a pyloric canal, which empties into the duodenum, the proximal segment of the small intestines. As mixing movements take place during digestion, the pylorus frequently changes shape. A muscular pyloric sphincter regulates the release of chyme into the duodenum. Glands in the pylorus secrete mucus and important digestive hormones, including gastrin, a hormone that stimulates gastric glands. The stomach's volume increases while you eat and then decreases as chyme enters the small intestine. When the stomach is relaxed or empty, the mucosa has prominent folds called rugae. These temporary features let the gastric lumen expand. The stomach can also stretch up to 50 times its empty size. As the stomach fills, the rugae gradually flatten out until a maximum distension they almost disappear. The world record set in 2013 for eating hot dogs with buns in 10 minutes is 69. When empty, the stomach resembles a muscular tube with a narrow constricted lumen. When full, it can contain 1 to 1.5 liters of material. The mucularis mucosae and mucularis externa of the stomach contain extra layers of smooth muscle cells in addition to the usual circular and longitudinal layers. The mucularis mucosa generally contain an outer circular layer of muscle cells. The mucularis externa has an inner, oblique layer of smooth muscle. The extra layers of smooth muscle strengthen the stomach wall and assist in the mixing and churning essential to the formation of chyme. Simple columnar epithelium lines all portions of the stomach. The epithelium is a secretory sheath, which produces a carpet of mucus that covers the interior surface of the stomach. The alkaline mucus layer protects epithelial cells against the acids and enzymes in the gastric lumen. Shallow depressions called gastric pits open onto the gastric surface. The mucus cells at the base, or neck, of each gastric pit actively divide, replacing superficial cells that are shed into the chyme. A typical gastric epithelial cell has a lifespan of 3 to 7 days. Exposure to alcohol or other chemicals that damage or kill epithelial cells increases cell turnover. In the fundus and body of the stomach, each gastric pit communicates with several gastric glands, which extend deep to the underlying lamina propria. Gastric glands are dominated by 2 types of secretory cells, parietal cells and chief cells. Together, they excrete about 1,500 milliliters of gastric juice each day. Parietal cells are especially common along the proximal portions of the gastric glands. These cells secrete intrinsic factor. This glycoprotein helps absorb vitamin B12 across the intestinal lining. Parietal cells also secrete hydrochloric acid. They do not produce hydrochloric acid in the cytoplasm, however. This acid is so strong that it would erode a secretory vesicle and destroy the cell. Instead, hydrogen and chloride, the 2 ions that form hydrochloric acid, are transported independently by different mechanisms. The initial step in HCl production is the formation of carbonic acid within parietal cells. The dissociation of carbonic acid releases bicarbonate and hydrogen ions. The hydrogen ions are actively transported into the lumen of the gastric gland. The bicarbonate ions are exchanged for chloride ions from the intestinal fluid. When gastric glands are actively secreting, enough bicarbonate ions diffuse into the bloodstream from the intestinal fluid to increase the pH of the blood significantly. This sudden influx of bicarbonate ions has been called the alkaline tide. The secretions of parietal cells can keep the stomach contents at a pH of 1.5 to 2. Although this highly acidic environment does not by itself digest chyme, it has four important functions. 1. The acidity of gastric juice kills most of the microorganisms ingested with food. 2. The acidity denatures proteins and inactivates most of the enzymes in food. 3. The acidity helps to break down plant cell walls and the connective tissues in meat. And 4. An acidic environment is essential for the activation and function of pepsin, a protein digesting enzyme secreted by chief cells. Chief cells, also called zymogenic cells, are the most abundant near the base of a gastric gland. These cells secrete pepsinogen, an inactive proenzyme. Acid in the gastric lumen converts pepsinogen to pepsin, an active proteolytic or protein digesting enzyme. Pepsin functions most effectively at a strong acidic pH of 1.5 to 2. In addition, the stomachs of newborn infants, but not of adults, produce renin, also known as chymosin, and gastric lipase. These enzymes are important for the digestion of milk. Renin coagulates milk proteins. Gastric lipase initiates the digestion of milk fats. Glands in the pylorus produce primarily a mucus secretion, rather than enzymes or acid. In addition, several types of enteroendocrine cells are scattered among the mucus-secreting cells. These enteroendocrine cells produce at least 7 hormones, most notably gastrin. Gastrin is produced by G cells, which are most abundant in the gastric pits of the pyloric antrum. Gastric stimulates secretion by both parietal and chief cells, as well as contraction of the gastric wall that mix and stir the gastric contents. The pyloric glands also contain D cells, which release somatostatin, a hormone that inhibits the release of gastrin. D cells continuously release their secretions into the intestinal fluid adjacent to the G cells. Neural and hormonal stimuli can override this inhibition of gastrin production when the stomach is preparing for digestion or is already engaged in digestion. Several other hormones play a role in hunger and satiety, which is a feeling of not being hungry. Levels of ghrelin, a hormone produced by P/D1 cells lining the fundic region of the stomach, increase before meals to initiate hunger. Ghrelin levels decrease shortly after eating to curb appetite. Ghrelin is also antagonistic to leptin, a hormone derived from fat tissue that induces satiety. Another hormone from the stomach and small intestine is obistatin, though it is thought to decrease appetite and inhibit thirst. The same gene encodes both ghrelin and obistatin. The production of acid and enzymes by the gastric mucosa can be 1. Controlled by the CNS, 2. Regulated by short reflexes of the enteric nervous system, coordinated in the wall of the stomach, and 3. Regulated by hormones of the digestive tract. Gastric control proceeds in 3 overlapping phases. They are named according to the location of the control center. You have the celiac phase, the gastric phase, and the intestinal phase. The stomach carries out preliminary digestion of proteins by pepsin. For a variable period, it allows the digestion of carbohydrates and lipids by salivary amylase and lingual lipase. These enzymes continue to work until the pH throughout the contents of the stomach falls below 4.5. They generally remain active 1 to 2 hours after a meal. As the stomach contents become more fluid and the pH approaches 2.0, pepsin activity increases. Protein disassembly begins. Protein digestion is not completed in the stomach because the time is limited and pepsin attacks only specific types of peptide bonds and not all of them. However, pepsin generally has enough time to break down complex proteins into smaller peptide and polypeptide chains before the chyme enters the duodenum. Although digestion takes place in the stomach, nutrients are not absorbed there. This is because 1. The epithelial cells are covered by a blanket of alkaline mucus and are not directly exposed to chyme. 2. The epithelial cells lack the specialized transport mechanisms of cells that line the small intestine. 3. The gastric lining is somewhat impermeable to water. And 4. Digestion has not been completed by the time the chyme leaves the stomach. At this stage, most carbohydrates, lipids, and proteins are only partially broken down. Some drugs can be absorbed in the stomach, though. For example, ethyl alcohol can diffuse through the mucous barrier and penetrate the lipid membranes of the epithelial cells. As a result, beverage alcohol is absorbed in your stomach before any nutrients in a meal reach the bloodstream. Meals containing large amount of fat slow the rate of alcohol absorption. Why is that? The reason is that alcohol is lipid soluble, and some of it will be dissolved in fat droplets in the chyme. Aspirin is another lipid-soluble drug that can enter the bloodstream across the gastric mucosa. Such drugs alter the properties of the mucus layers and can promote epithelial damage by stomach acid and enzymes. Prolonged use of aspirin can cause gastric bleeding, so individuals with stomach ulcers usually avoid aspirin.